Okay, welcome back from the poster session. I'm Sally Permar, and I'm the co-leader of the Development Corps with Herman Stats. So come talk to me if you have an idea for a small grant or want to be an early career grant reviewer. We're always looking for those. But this is going to be an exciting session where we're going to be hear presentations that were selected from the abstract pool. And so we have three very different areas of HIV research that I think really reflects the work that Duke is doing under the CFAR. So our first speaker is Wilton Williams. He's a postdoctoral fellow working in the lab of Bart Haynes and Georgia Tamaris and at the Human Vaccine Institute. He's originally from Kingston, Jamaica, and received his doctorate degree from the University of Florida. And he is focusing his research on HIV immunobiology and vaccine development, and has recently had some major findings that were published in Science last month that he's going to tell us about that have major impacts on the direction for HIV vaccine development. So thank you, Wilton. Sorry.
Thank you, Dr. Permar. We previously observed that acute HIV-1 infection induced a dominant GP41 reactive plasma and memory B-cell antibody response. Additionally, the GP41 reactive antibodies in acute and chronic HIV infection were polyreactive for host and intestinal microbiota antigens. The HIV Vaccine Trials Network Phase 2b trial, or HVTN 505, recently studied the VRC's DNA prime recombinant adenovirus serotype 5 boost, or AD5 vaccine regimen in AD5 seronegative circumcised males in the US. While the HVTN 505 trial showed no vaccine efficacy, of interest to our study was that the HVTN 505 was the first efficacy trial with envelope GP41 in the immunogen. The purposes of our study were 1, to determine if VRC DNA prime AD5 boost HIV-1 vaccine induces a dominant GP41 reactive antibody response, and 2, to determine if the antibodies induced by the VRC DNA prime AD5 boost vaccine were polyreactive to host antigens, including intestinal microbiota. The multiclade prime boost VRC vaccine contained a DNA prime of subtypes A, B, and C envelopes, and subtype B GAG, POL, and NEF. The boost contained AD5 that expressed matching envelopes used in the prime and a GAG, POL fusion protein. The prime was administered monthly for the first 3 months, and the boost followed at month 6 after the initial prime. We studied antibodies present at 4 weeks post-AD5 boost from participants in the Phase 1b HVTN-082, Phase 2 HVTN-204, and Phase 2b HVTN-505 trials. The HVTN-082, 204, and 505 trials all studied the VRC vaccine. The methods used in our study included isolation of antibodies from HIV-1 envelope-reactive memory B cells via antigen-specific B cell sorts. Isolated antibodies were characterized for binding specificities and function. The antigens used as hooks for our memory B cell sorts included HIV-1 group M consensus or M.CONS GP140, as well as the vaccine-matched GP140 and V1V2 fusion proteins. First, we studied the plasma and memory B cell repertoire induced by the VRC DNA prime AD5 boost HIV-1 vaccine. Plasma at 4 weeks post-AD5 boost from 40 randomly selected HVTN 505 trial participants who were HIV-1 negative at final month 24 visit was screened for binding to MNGP41 recombinant protein, as well as vaccine-matched and CONS GP120 envelope in a Luminex binding assay. Envelope oligomers are shown on the x-axis, and binding antibody titer is shown as area under the curve on the y-axis. The higher the area under the curve, the better the binding. As you can see, plasma-binding IgG antibody titers for vaccine responders, shown in red circles, were significantly higher for MNGP41 reactivity than for binding to GP120 envelopes, including the vaccine strain envelopes. From 8 HVTN-082 and 204 trial participants who had high plasma antibody titers to vaccine strain GP140 envelopes, we also observed significantly higher plasma-binding IgG antibody titers for MNGP41 reactivity than for GP120 envelopes, including the vaccine strain envelopes. From 8 HVTN-082 and 204 trial participants, I isolated 221 envelope-reactive antibodies from blood memory B cells. Of the 221 envelope-reactive antibodies, 93% targeted envelope GP41. In contrast, 7% of the envelope-reactive antibodies targeted envelope GP120. Next, we asked the question, what is the predominant variable heavy chain family usage of the VRC vaccine-induced GP41 reactive antibodies? Of 205 GP41 reactive antibodies, 61% used VH169 gene segment, compared to HIV-1 uninfected individuals, where only 6% of the antibody repertoire used VH169. Previous observations have taught us that VH169 gene segment has 13 alleles separated into two groups defined by the amino acid at HCDR2 position 54. 54F alleles encode a phenylalanine at HCDR2 position 54, whereas 54L alleles encode a leucine at HCDR2 position 54. It is important to note that HIV-1 GP41 reactive neutralizing antibodies D5 and HK20 and influenza stem broadly neutralizing antibodies use 54F VH169 alleles. Thus, we next ask the question, what are the VH169 allelic variants used for HIV-1 infected compared to the VRC vaccine-induced GP41 reactive antibodies? We evaluated the frequency of HCDR2, 54L, and 54F amino acid variants of VH169 GP41 reactive antibodies isolated from 8 HVTN 082 and 204 trial participants. Of 125 VH169 using GP41 reactive antibodies that we could unambiguously classify as 54L or 54F amino acid variants, 93% used 54L amino acid variants, and only 7% used 54F amino acid variants. For comparison, we next evaluated the frequency of 54L and 54F amino acid variants of VH169 using GP41 reactive antibodies induced via HIV-1 infection. Of 42 VH169 using GP41 reactive antibodies isolated from HIV-1 infected subjects that we could unambiguously classify as 54L or 54F HCDR2 variants, 98% used 54L amino acid variants, in contrast to 2% that used 54F amino acid variants. However, of 64 VH169 non-HIV-1 reactive antibodies isolated from HIV-infected subjects, 72% used 54F amino acid variants, while 28% used 54L amino acid variants. These data demonstrated that the VRC vaccine preferentially elicited VH169 using GP41 reactive antibodies of HCDR2 54L amino acid variants consistent with HIV-1 infection. In contrast, VH169 using non-HIV-1 reactive antibodies preferentially use 54F amino acid variants. Next, we characterize the clonal lineages of VRC vaccine-induced GP41 reactive monoclonal antibodies, or MOABs, for binding specificities and functions. 17 GP41 reactive antibodies representative of different clonal lineages were generated as purified MOABs and functionally characterized. All 17 GP41 reactive MOABs bound recombinant MNGP41 protein, and 16 of 17 bound post-fusion forms of GP41 proteins. While none of the GP41 reactive MOABs bound MPER peptides targeted by GP41 broadly neutralizing antibodies, 3 representative MOABs of highest GP41 reactivity demonstrated binding to GP140 enzyme-digested fragment and linear peptides containing the peptide repeat 1 or HR1 sequence LLRAIE. The GP41 reactive MOABs tested were non-neutralizing and only 1 weakly captured infectious HIV-1 strains or mediated ADCC against HIV-1 infected cells. Since we previously found GP41 reactive antibodies in acute and chronic HIV infection to be polyreactive for host and intestinal microbiota antigens, including the 37 kilodalton subunit of E. coli RNA polymerase, we next tested the 17 vaccine-induced GP41 reactive MOABs for binding aerobe and anaerobe ulcer lysates of intestinal microbiota and E. coli RNA polymerase. GP41 microbiota lysates and E. coli RNA polymerase are shown on the x-axis, while binding is shown as mean fluorescence intensity, or MFI, on the y-axis. The higher the MFI, the better the binding. Approximately 80% of MOABs bound intestinal microbiota lysates, and 47% bound recombinant E. coli RNA polymerase. Additionally, Western blot and mass spectrometry analyses demonstrated that GP41 reactive MOABs cross-react with a second microbiotic protein candidate called pyruvate flavodoxine oxidoreductase, which shares a sequence similarity with GP41. Pyruvate flavodoxine oxidoreductase contains LLR-GIK sequence that is similar to ALR-AIL sequence in E. coli RNA polymerase subunit and the GP41HR1 sequence LLR-AIE. Furthermore, 65% of GP41 reactive MOABs bind autoantigens, including DNA and nuclear proteins, and 29% of GP41 reactive MOABs bind Ep2 cells. These data demonstrated that the DNA prime Ad5 boost vaccine-induced GP41 reactive MOABs bound intestinal microbiota antigens and are polyreactive, consistent with GP41 reactive antibodies induced by HIV-1 infection. To determine the origin of the post-vaccine GP41 intestinal microbiota cross-reactive antibodies, we asked if we could isolate GP41 intestinal microbiota cross-reactive antibodies from pre-vaccination antibody repertoire of VRC vaccine recipients. To answer this question, we performed next-generation sequencing of variable heavy chain repertoire in duplicates of pre-vaccine RNA samples from blood of 8-HVTN 082 and 204 trial participants. We expressed and characterized clonally related pre- and post-vaccine antibodies by complementing the pre-vaccine heavy chain with the post-vaccine antibody light chain. This slide shows the clonally related pre-vaccine and post-vaccine VH chains, isolated from a vaccinee. The clonally related pre- and post-vaccine VH chains share the same VDJ recombination and HCDR3 length, and the HCDR3s shared greater than 80% nucleotide sequence identity. While the pre-vaccine VH chain was an unmutated IgM, the post-vaccine VH chain was 5.3% mutated IgG. Next, we tested the binding specificities of the clonally related pre-vaccine IgM antibody DH477 and the post-vaccine IgG antibody DH476. The antibodies were tested for intestinal microbiota and envelope GP41 reactivities via SPR. Both the pre-vaccine antibody DH477 and post-vaccine antibody DH476 bound intestinal microbiota antigens and GP41 proteins. However, intestinal microbiota reactivity decreased from pre-vaccination antibody DH477 to post-vaccination antibody DH476, while in contrast, relative binding affinity to the post-fusion forms of GP41 increased from pre-vaccination antibody DH477 to post-vaccination antibody DH476. In summary, like AHI, vaccination with a GP41-containing vaccine induced a dominant GP41 reactive plasma and memory B-cell antibody response. The GP41 reactive antibody response was non-neutralizing and cross-reactive with intestinal microbiota and to host antigens. The GP41 reactive antibody response was VH169 dominant and restricted to the 54L HCDR2 variants. Pyruvate flavodoxine oxidoreductase was a second candidate microbiota protein that cross-reacted with GP41 reactive antibodies. And using next-generation sequencing, we demonstrated the presence of a pre-vaccination GP41 intestinal microbiota cross-reactive B-cell that gave rise to a post-vaccination GP41 intestinal microbiota reactive antibody. In conclusion, we have demonstrated that a dominant polyreactive GP41 antibody vaccine-induced response can originate from a pool of GP41 intestinal microbiota cross-reactive pre-vaccination B cells. These data raise the hypothesis that if these results can be shown in other vaccine trials, one region of GP41 in vaccine immunogens that might be considered for deletion or modification to avoid intestinal microbiota GP41 cross-reactivity includes amino acids in the GP41 HR1 region. I would like to end by acknowledging my mentors, Drs. Haynes and Tamaris, members of the DHVI team, our collaborators, and the vaccinees. Finally, many thanks to our funding agencies for the invitation to speak today and for your attention. Thank you.
So every antibody that's elicited by a vaccine has to originate from a B cell that existed pre-vaccination, right? So the question is, how do you distinguish the fact that the possibility that this arose from a naive B cell, which would be an IgM unmutated ancestor, versus a B cell that was elicited by the microbiota?
Well, so for that question, so while a great majority of the naive B cells are IgM positive, IgD positive phenotype, there is a minority that are also unmutated, the IgM positive, IgD negative, but we're not able to distinguish which one of those populations of or antibodies are coming from. So that's the answer to that in all regards. Based on CD4 T cells, we know Mark Davis's group at Stanford has shown that in virus-naive individuals, you can isolate CD4 T cells that are HIV reactive that are also stimulated by microbiota peptides. And what they've shown is that in adults, the phenotype of those T cells are memory based on the CD45 or O expression, but in infants, based on cord blood, those are naive. So there seems to be the early onset beginning with the naive, and then at some point this differentiates in the memory T cells, which occurs after birth but before adulthood. So in the case of B cells, something similar could be going on.
Right, and I agree. I think it's important, though, that you try to establish whether or not those clones showed any evidence of pre-existing expansion or differentiation.
Correct. Thank you.
Other questions for Wilton? So just one question. You talked about a strategy to get around maybe a pre-existing B-cell response for vaccines would be deleting a region that's immunodominant in the pre-existing repertoire, but are there other strategies that we might be able to use to, make better vaccines?
Oh, yeah, certainly. One way would be to prime with the GP120s. So you bring out the B cells that are GP120 reactive and give those a competitive advantage or give them an opportunity to compete with the already pre-existing GP41 reactive antibodies, and then you boost with a GP140. So at that point, it's an equal race between the 41s and the 120 B cells.
Okay, great. Thank you, Wilton.
Thank you.